Hello and welcome, dear students. Today we are going to discuss about Mendelian inheritance, that is single factor, multifactorial inheritance and polygenic inheritance. The main objectives of today's lecture are to know about the Mendelian single factor and their inheritance patterns, to know about the multifactorial inheritance pattern and the complex diseases, to understand the characteristics of multifactorial diseases, and finally to understand about the diabetes mellitus that shows the complex traits. Dear students, let's first start with the introduction. A Mendelian trait is one that is controlled by a single locus in an inheritance pattern. In such cases, a mutation in a single gene can cause a disease that is inherited according to Mendel's principles of inheritance. Dominant diseases manifest in heterozygous individuals, while as recessive ones are sometimes inherited unnoticeably by genetic carriers. Examples include sickle cell anemia, Tay-Sachs disease, cystic fibrosis, and xeroderma pigmentosa. A disease controlled by a single gene contrasts with a multifactorial disease, like heart disease, which is affected by several loci and the environment, as well as those diseases inherited in a non-Mendelian fashion. Mendel explained inheritance in terms of discrete factors, now called as genes, that are passed along from one generation to generation according to the rules of probability. Mendel's laws are valid for all sexually reproducing organisms, including garden peas and human beings. However, Mendel's laws stop short of explaining some patterns of genetic inheritance, as most often the inheritance patterns are more complex. Mendel chose genetic traits in plants that are determined by only two alleles, such as allele dominant allele A and recessive allele A. In nature, genes often exist in several different forms with multiple alleles, as that in human blood group. Furthermore, many traits are produced by the interaction of several genes together. Traits controlled by two or more genes are said to be polygenic traits. Any human trait requires the performance of many genes and environmental factors. The few single characters are said to depend on the particular genotype at the single gene locus. These characters expressed by the only pair of genes are called as the Mendelian characters. Pedigree analysis can give different patterns of inheritance for the Mendelian traits. There are thousands of Mendelian characteristics found in humans. Single gene diseases are usually inherited in one of several patterns depending on the location of gene and whether one or two normal copies of the gene are needed for the disease phenotype to manifest. Apart from the single gene characters, there are many characters that can be associated with many genes at different locus. For any DNA variants, the gene affected is the primary reason, and it is more likely the Mendelian. The basic laws of inheritance are important in understanding patterns of disease transmission across generations. Most genes have one or more versions due to mutations or polymorphisms referred to as alleles. Individuals may carry a normal allele and or a disease or rare allele depending on the impact of the mutation or polymorphism and the population frequency of the allele. There are 5 basic modes of inheritance for single gene diseases. 1. Autosomal dominant. 2nd. Autosomal recessive. 3rd. X-linked dominant. 4th. X-linked recessive, and the 5th one is mitochondrial. Genetic heterogeneity is a common phenomena with both single gene disease and complex multifactorial diseases. It should not be surprising that multiple affected family members may experience different levels of disease severity and outcomes. But in case of protein level of variants, like functions of proteins can depend on more than one gene locus. This can be because of the post-translational modifications. Some of the defects are seen in the early age of birth as congenital heart disease resulting from defects in many factors. Hence, these are under multifactorial defects. The characters depending on many genes at different loci are known as the non-Mendelian characters. They can also be termed as multifactorial, where it states that the many factors for a single defect. Genetic heterogeneity is a common phenomena with both single gene disease and complex multifactorial diseases. Hence, in this lecture, the following aspects are going to be discussed. First, the Mendelian single factor characters and their inheritance patterns. Mendelian inheritance refers to the inheritance of traits controlled by a single gene with two alleles, one of which may be completely dominant to the other recessive one. The pattern of inheritance of Mendelian traits depends on whether the traits are controlled by genes on autosomes or by genes on sex chromosomes. Two tools for studying inheritance are the pedigree analysis and the Punnett squares. A pedigree is a chart that shows how a trait is passed from one generation to the next generation within a family. A Punnett square, on the other hand, is a chart that shows the expected ratio of possible genotypes in the offspring of two parents when they are crossed with each other. Examples of human autosomal Mendelian traits include albinism and Huntington's disease. Examples of human X-linked traits include red-green color blindness and hemophilia. Dear students, now let us discuss the autosomal recessive single gene disease. Autosomal recessive single gene disease occur only in individuals with two mutant alleles of the disease-associated gene. Remember, for any given gene, a person inherits one allele from his or her mother and one allele from his or her father. Therefore, individuals with an autosomal recessive single gene disease inherit one mutant allele of the disease-associated gene from each of their parents. In pedigrees of families with multiple affected generations, autosomal recessive single gene diseases often show a clear pattern in which the disease skips one or more generations. Dear students, let's now discuss the phenylketonuria disease, which is a prominent example of a single gene disease with an autosomal recessive inheritance pattern. Phenylketonuria is associated with mutations in the gene that encodes the enzyme phenylalanine hydroxylase, or PAH. When a person has these mutations, he or she cannot properly manufacture phenylalanine hydroxylase enzyme. So he or she is subsequently unable to break down the amino acid phenylalanine, which is an essential building block of dietary proteins. As a result, individuals with phenylketonuria accumulate high levels of phenylalanine in their urine and blood, and this build-up eventually causes mental retardation and behavioral abnormalities. Let's discuss the other examples of single gene disease with an autosomal recessive inheritance pattern, that is albinism. Albinism is associated with autosomal recessive mutations in the OCA2 gene. This gene is involved in biosynthesis of the pigment melanin, which gives color to a person's hair, skin, and eyes. The condition called albinism results from a lack of pigment melanin in the skin, hair, and eyes. albinism is usually controlled by a single autosomal gene with two alleles. The allele for normal pigmentation, let's call it R, capital R, is dominant to the allele for albinism, that is small r. Individuals with either an RR, that is capital RR, or capital R small genotype will not have albinism, because the R, which is dominant allele, is dominant over the recessive small r allele. However, consider what happens if two individuals with the heterozygous condition, that is capital or small r genotype, reproduce with each other. Their possible offspring could be capital R, that is normal pigmentation, capital R, small r, that is normal pigmentation, or small r, that is a recessive condition, it is albinism. This explains why a child with albinism, that is small r recessive genotype, can have two parents that do not have albinism in them. Both unaffected parents must be a carrier of the recessive R allele, but they also have a dominant R allele that prevents them from having the condition themselves. So they are albinism free. Several other human diseases, including cystic fibrosis, sickle cell anemia, also exhibit an autosomal recessive inheritance pattern. Cystic fibrosis is associated with recessive mutations in the CFTR gene, where sickle cell anemia is associated with recessive mutations in the beta hemoglobin gene. Dear students, let us now discuss the autosomal dominant single gene diseases. Autosomal dominant single gene disease occur in individuals who have a single mutant copy of the disease associated gene. In this case, the presence of a single non-mutant or wild type copy of the gene is not enough to prevent the disease. Individuals can inherit the mutant copy of the disease associated gene from either an affected mother or an affected father. Huntington's disease is a progressive neurodegenerative disorder and is a well-known example of an autosomal dominant single gene disease. Most individuals with a single copy of the mutant Huntington gene, that is HTT, will have Huntington disease later in the life. Typically, autosomal dominant diseases affect individuals in their early years and prevent them from living past infancy or childhood, which in turn precludes these individuals from reproducing and potentially passing on the mutation to other offsprings. In the case of Huntington's disease, however, the late onset of the disorder means that many affected individuals have already had children before they are even aware that they carry the mutation. Disease-associated change in the Huntington's gene consist of a special type of mutation called triplet repeats. These mutations are simply extra repetitions of the 3 bases in DNA sequence CAG. The number of CAG repeats in a mutated Huntington gene determines the age at which a person will develop Huntington's disease, as well as how severe the condition will be. Genetic tests can be used to determine how many CAG repeats are in an individual's Huntington's gene, thereby providing a highly accurate assessment of the individual's disease risk. Because affected parents have a 50% chance of passing a mutant copy of the Huntington gene onto each of their offspring, children of people with Huntington's disease are often faced with the dilemma of whether to undergo such testing. Genetic testing can either provide immediate relief in knowing that one is free from the disease, or the confirmation that one will certainly suffer from the condition at some point in the future. Dear students, let's now discuss X chromosome linked recessive single gene diseases. Single gene diseases that involve genes found on the sex chromosomes have somewhat different inheritance patterns than those that involve genes found on a person's autosomes. The reason for these differences lies in the genetic distinction between males and females, because males have one X chromosome while females have two. Recall that the females have two copies of the X chromosome and they receive one copy from each parent. Therefore, females with an X chromosome linked recessive disease inherit one copy of the mutant gene from affected father and the second copy of the mutant gene from their mother, who is most often a carrier that is in heterozygous condition, but who might be affected, that is homozygous. Males, on the other hand, have only one copy of X chromosome which they always receive from their mothers. Therefore, males with an X chromosome linked disease always receive the mutant copy of the gene from their mother. Moreover, because men don't have a second copy of the X chromosome to potentially cancel out the negative effects of X-linked mutations, they are far more likely than women to be affected by X chromosome linked recessive diseases. The blood clotting disorder hemophilia A is one of several single gene diseases that exhibit an X chromosome linked recessive pattern of inheritance. Males who have a mutant copy of the factor 8 gene, that is F8, will always have hemophilia. In contrast, women are rarely affected by this disease, although they are most often carriers of the mutated gene. Duchenne muscular dystrophy, or DMD, is another example of a single gene disease that exhibits an X chromosome linked recessive inheritance pattern. This condition is associated with mutations in the dystrophin gene itself. Dear students, let's now discuss the X chromosome linked dominant single gene diseases. Few dominantly inherited forms of human diseases are X chromosome linked. Females with an X chromosome linked dominant disease can inherit the mutant gene from either an affected mother or an affected father, whereas males always inherit such diseases from an affected mother. Examples of X chromosome linked dominant disease are rare, but several do exist. For instance, dominant mutations in the phosphate regulating endopeptidase gene PHEX, which resides on the X chromosome, are associated with X-linked dominant hypophosphatic rickets. Similarly, Rett syndrome, a neurodevelopmental disease, is associated with dominant mutations in the methyl CpG binding protein 2 gene, that is MECP2. Rett syndrome almost exclusively affects females because male embryos with a dominant mutation in the MECP2 gene rarely survive. Dear students, now let us discuss the Y chromosome linked single gene diseases. Like X-linked dominant disease, Y chromosome linked disease are also extremely rare, because only males have a Y chromosome and they always receive their Y chromosome from their father. Y-linked single gene diseases are always passed on from affected fathers to their sons. It makes no difference whether the Y chromosome linked mutation is dominant or recessive, because only one copy of the mutated gene is ever present, and that too is in the father. Thus, the disease-associated phenotype always shows. One example of a Y-linked disorder is non-obstructive spermatogenic failure, a condition that leads to infertility problems in males. This disorder is associated with mutations in the ubiquitin-specific protease 9-1 gene, that is USP9Y, on the Y chromosome. Dear students, this was all about single factorial characters and their inheritance pattern. Now let us discuss the multifactorial characters and inheritance pattern of multifactorial diseases. After the rediscovery of the Mendelian works in 1900 by Hugo de Vries, Karl Correns, and Teschenmark, many of the biometricians agreed for the Mendelian pattern of inheritance in case of few abnormalities. But they also disagreed when it came to the characters like height and size of the body, besides many others. These controversies continued till 1918 until the demonstration of R.A. Fisher about the characters which are ruled by many independent Mendelian factors. It was termed as the polygenic characters. Polygenic characters are the characters that are associated with many genes and are continuous variation that occurs in generations. They inherit in a non-Mendelian fashion. Many polygenic traits get influenced by the other external factors along with the polygenes. External factors can be the environment, drugs, etc. Hence, it's called as the multifactorial inheritance of the polygenic traits. So polygenic traits include height, behavior, and other disease conditions like club foot formation, cleft lip, diabetes mellitus, and hydrocephalus. In most of the cases of multifactorial conditions, the number of genes involved is not precisely known. The characters like the formation of cleft lip or cleft plate will be associated with many syndromes and result by the chromosomal abnormalities, few mutations, and other environmental factors. Both traits of cleft lip and cleft plate can be seen in a single person. Some genes can be responsible for the trait cleft lip, and some other genes can cause cleft palate. Both traits can be seen in males more often than females. The genetic and multifactorial conditions can influence both characters. Females can suffer from rear cleft palate conditions more than males. In the case of cleft lip and cleft plate, both genetic and environmental factors will play an essential role in the development of the phenotype. These traits will develop at birth. The children born with this defect will not show the affected parents and any other family history of being affected. Parents can contribute to this defect by some non-functional genes. Hence, it shows that the parent who do not show any variation will not reach the threshold point. They will not develop the trait. The affected individual will have the factors which are contributing to this trait will reach the threshold point. In the increased condition of the threshold, the trait will be more severe. Hence, cleft lip and cleft palate come under the multifactorial diseases with discontinuous variation. Dear students, let's now discuss the inheritance of multifactorial traits, that is the polygenic inheritance. For years, followers of Mendel and Galton argued over whether the Mendelian theory or the Galtonian theory correctly describes inheritance in humans. Although Mendelian inheritance correctly described some human diseases and traits, it did not describe all of them. Likewise, the Galtonian theory did not fit all observations. Today, we know that both theories correctly describe inheritance patterns for different traits, including various human diseases. The question of whether a disease exhibits Mendelian or Galtonian patterns simply depends on which disease we are studying and talking about. The multifactorial diseases will not show Mendelian pattern of inheritance. Environmental factors will influence the disease and they can increase or decrease the risk of that particular disease. These diseases can affect anyone's gender more often than another gender. It will not show any sex-limited trait. The relatives of the affected individuals are more susceptible to getting those traits. Some of the diseases will fall into a discrete condition and they are discontinuous. Some other diseases will show a continuous development of the phenotypes in the patterns of gradient and are ongoing characters. In some cases, the phenotype of discontinuous characters resembles the phenotypes of continuous variation. This shows that continuous variation will have some threshold for the disease to show their frequency of development. Infection can occur and develop in a state of the higher limit. If the variation is limited and not reach up to a threshold point, then the disease will not develop. They will be in a discontinuous condition. The multifactorial disease will show the discontinuous variation. There are many diseases which show the multifactorial conditions. They include multiple sclerosis, diabetes mellitus, cancer, and asthma. The copy number variations or CNVs, epistatic change, and several environmental factors will contribute to the multifactorial diseases. There are many methods for studying common multifactorial diseases. One such way is the genome-wide association study, which helps in the identification of the common alleles which are responsible for the complex disorders. Dear students, let's now discuss the characteristics of a multifactorial disease. A multifactorial disease has a combination of distinctive characteristics that can be differentiated from clear-cut Mendelian or sex-limited conditions. These traits include: the disease can occur in isolation, with affected children born to unaffected parents. Although familial aggregation is also common, that is, there may be multiple cases in the same family, there is no clear Mendelian pattern of inheritance. Environmental influences can increase or decrease the risk of the disease. The disease occurs more frequently in one gender than the other, but it is not a sex-limited trait. In addition, first-degree relatives of individuals belonging to more rarely affected genders have a higher risk of bearing the disease. The concordance rates in monozygotic and dizygotic twins contradict Mendelian proportions. A concordance rate is a measure of the rate at which both twins bear a specific disease. The disease occurs more frequently in a specific ethnic group, that is, Caucasians, Africans, Asians, and Hispanics, etc. Going back to the example of coronary artery diseases, we know that there are a number of factors that increase the risk of disease onset, including obesity, type 2 diabetes, high blood pressure, high levels of low density lipoprotein cholesterol, and even gum diseases. Although coronary artery diseases runs in families, it does not show Mendelian pattern of inheritance and can occur in isolation. Coronary artery diseases also occurs more often in men than women, and its risk is higher among African Americans than among Caucasians or Asians. All of these characteristics are consistent with the classification of coronary artery diseases as a multifactorial disorder. Dear students, let's now discuss the common multifactorial disease, that is the diabetes mellitus complex trait. Multifactorial inheritance refers to the disorders and genetic traits that occur and are determined by the interaction of environmental factors and multiple genes. Multifactorial inheritance generally is thought to encompass threshold traits, qualitative traits, and complex disorders of adulthood, such as diabetes and heart diseases. Diabetes mellitus is a common multifactorial disorder where the body cannot produce enough amount of insulin. The carbohydrates from the food will get metabolized to give glucose and other molecules. This extra glucose will be taken up by the insulin and it will maintain the glucose level in the blood between normal range of 80 to 120 deciliter per cubic millimeter of the blood. When there is no production of insulin, then there will be problems in the metabolism of carbohydrates, lipids, and proteins. The lack of insulin will increase the amount of glucose in the blood and tissues. Increased blood glucose levels result in the diabetes mellitus. Insulin is produced by an organ called the pancreas. There are islets of Langerhans in the pancreas which contain beta cells in this organ which instructed to produce insulin and release them to the bloodstream. When we eat food for energy, it contains more amounts of carbohydrates. Intake of food will automatically increase blood glucose levels. The insulin will play a role in that situation. Insulin released into the bloodstream will make use of the excess glucose from the blood, and that will help in the storage of that glucose in the form of energy in the liver, that is glycogen. It helps during the low blood glucose levels. There are mainly two types of diabetes mellitus, and they are type 1 diabetes, that is insulin dependent diabetes, and type 2 diabetes, that is non-insulin dependent diabetes. Type 1 diabetes occurs in childhood, hence it's also termed as the juvenile form. This type will affect the children at a very young age. There is a high risk of other children in the family that gets affected by this condition, since this type of insulin dependent diabetes requires insulin intake for survival. The autoimmune condition of the body can cause this disease. In the cases of autoimmune conditions, the immune system will affect our body cells or organs. In type 1 diabetes, the immune system will affect the pancreatic cells and damage them. This will affect the production of insulin in the body. Type 2 diabetes is a multifactorial disease caused by both oligo and polygenic genetic factors as well as non-genetic factors that result from a lack of balance between the energy intake and the output and other lifestyle related factors. Type 2 diabetes is a common form of diabetes mellitus. This condition will occur in the old age instead of happening at a young age. Hence, this type can also be termed as the late onset type or the adult onset type. In this condition, the pancreas can produce a small amount of insulin which is not enough for the proper functions. In some conditions, the other cells of the body develop into resistance for the intake of insulin. So unlike type 1 diabetes, in this condition, people of the family are at less risk of developing this type 2 condition. It also depends on the environment they live and the type of food they eat. Even though type 2 is the milder form, it can lead to some other health problems if it gets severe in the blood vessels of the eyes, kidneys, and some heart problems. Diabetes mellitus has no cure and it can only be prevented in daily life. People with diabetes are often guided to eat a proper diet which has more fibers and to do exercise which can reduce fat formation. The affected people are more prone to infections and the healing of wounds takes many days. In conclusion, the condition for the multifactorial inheritance are genes and the environment together. Diabetes mellitus is another multifactorial condition that affects other organs of the body, which is more common in current days. Dear students, it was all about today's lecture regarding Mendel's inheritance, single factor versus multifactorial inheritance. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.